thank you for standing by. Today's call is being recorded. If you have any objections, you may disconnect at this time. All participants are in a listen-only mode until the question and answer session of today's conference. At that time, you may press star one on your phone to ask a question. I would now like to turn the call over to your host, Josh Finch. You may begin.
Good afternoon. I'm Josh Finch from NASA's Office of Communications. Thank you for joining us for today's call with experts to talk about science investigations launching on SpaceX's 26th commercial resupply mission to the International Space Station. The mission will launch on the Dragon spacecraft and Falcon 9 rocket from Launch Complex 39A at NASA's Kennedy Space Center in Florida. Here to talk more with us about some of the research launching aboard the SpaceX Dragon, we have Dr. Kurt Costello, Chief Scientist for the International Space Station Program and Research Integration Office Deputy Manager, who will discuss the breadth of research being done aboard the orbiting laboratory. Next, we'll have Dr. Timothy McCauley, Cypher Vestibular Health Study Lead, who will discuss the Falcon goggles. Dr. Joy Amasa, NASA Spacecraft Production Scientist, who will discuss the VEG-05 study. Matt Nickel, Boeing Development Project Senior Manager, who will discuss the next steps of the International Space Station Rollout Solar Arrays, or IROSAs. Myra Nelman-Gonzalez, Immunologist, and Johnson Space Center's Immunology and Virology Laboratory, who will discuss the Moon Microscope study. We have John Hogan, NASA Ames Research Center Bioengineering Branch Chief and Bionutrients 2 Principal Investigator. And then we have Martin Nisser, PhD candidate at the Massachusetts Institute of Technology and Science Lead for the Extrusion Project. Our speakers will provide a brief overview of their research, and then we'll have a time for questions after each speaker. So as a reminder, you can press star one to enter the queue after each speaker. First, we'll hear from Dr. Kurt Costello, Chief Scientist from the International Space Station. Dr. Costello?
Thanks, Josh. At NASA today, we've had a successful berthing of the Northrop Grumman 18 Cygnus cargo vehicle. Now we're eagerly awaiting the arrival of SpaceX's 26th commercial resupply mission. The International Space Station is an amazing multidisciplinary lab offering unique access to long-duration microgravity and the low Earth orbit space environment. Each CRS mission brings a wealth of new research to the station. SpaceX missions are specifically helpful in their ability to return samples developed in space to researchers on the ground for further analysis. Several of our researchers will be joining us today with details on their research. The research highlighted today will add to the over 3,400 investigations already conducted on the ISS and focus on medical technologies and capabilities that will enable NASA's Artemis exploration mission. It will help us to enhance our ability to grow food on orbit and to build new structures in space. The SpaceX 26 Dragon vehicle also carries two IROSA, or ISS rollout solar arrays, that will be attached and augment the power generation capability of the ISS to keep us operating and expand our capabilities into the next decade. We're very thankful to have our researchers with us to explain their research in their own words today. Back to you, Josh.
Thank you, Dr. Costello, for that welcome. And we'll now hear from our first researcher as a part of today's call. And I'll start with Dr. Timothy McCauley.
Okay, thanks, Josh. So the Falcon goggles are the hardware that are being sent up, and basically they look and fit like a scuba mask, but they're a lightweight system used for video oculography, which is a non-invasive video-based method of tracking eye movements using small cameras in the goggles, and they have inertial sensors as well to track head movements. So the goggles will be used for vestibular exams as part of our Cypher Vestibular Health Study. Cypher is a complement of integrated research studies, and vestibular health is our specific project in the JSC Neuroscience Lab. I want to tell you a little bit more about this study. We know that there are significant vestibular and sensory-motor adaptations during spaceflight, especially around gravity transitions, like when first going to space and when landing back on Earth. and these can cause astronauts to experience motion sickness and deficits in both perceptual and motor functions. These are normal adaptations, but if the extent and time course of these changes increase with flight duration, then they can impact astronauts' abilities to perform critical mission tasks during future deep space exploration. So the goal of the study is to perform well-established clinical and experimental procedures with the Falcon goggles to assess astronauts' vestibular health and performance before, during, and after spaceflight. And these exams will involve ocular alignment measures and eye and head movement measures while the astronauts actively move in specific patterns and while the astronaut is passively moved by one of their crew members in specific patterns. By doing this, we can identify the timeline trends in vestibular adaptation and understand how the vestibular system is adapting. And then ultimately, this will help us develop countermeasures to help astronauts with their vestibular health and performance in flight and post-flight. With that, I'm happy to answer any questions, Josh.
Thank you. We'll now take questions for Dr. McCauley. I'll pause here for give time to reporters to press *1 if you have a question for this investigation. And while we wait, I wondered if you might be able to talk a little bit more about how this study will help in the development of countermeasures.
Yeah, sure thing. So, this study will help us determine which countermeasures might be helpful and when. Depending on how the vestibular system adapts to long-duration spaceflight, we can propose countermeasures based on the vestibular rehabilitation therapies that are currently used in patients with vestibular disorders. So we definitely collaborate with clinical, doctors that work with patients that have vestibular loss, both peripheral and, lateral on one side. So we'll work with them to determine the best countermeasures.
Thank you. And at this time, we have no questions in the queue. We will come back around toward the end. So if any reporter has a question that didn't get to press *1 in time, we will come back. But next, we'll hear from Dr. Joy Amasa.
Okay, thank you. So plants are a nutritious, palatable food source that we can grow for the crew during spaceflight. And this has the potential to promote health, provide bioavailable nutrients, enhance the dietary experience and reduce things like menu fatigue, and reduce launch mass moving forward for longer duration exploration missions. But studies of edible produce being grown during spaceflight have been pretty limited, and so we have a knowledge gap in methods to grow safe, acceptable, nutritious crops. So we are using the vegetable production system called Veggie on the International Space Station as an opportunity to look at developing pick-and-eat components to the space diet as a first step for this bioregenerative supplemental food production. So this is a grant called Pick-and-Eat Salad Crop Productivity, Nutritional Value, and Acceptability to supplement the ISS food system, which is leading to the SEDGE-05 investigation. And this is a hybrid investigation. So we have plant research as well as human research, organoleptic and behavioral health research. This is the second half of an investigation which began with Veg-04 in 2019, growing Mizuna mustard, a leafy green crop, and Veg-05 will grow red robin dwarf tomatoes. This research is sponsored by NASA's Human Research Program and implemented in partnership with NASA's Space Biology Program. And our Veg-05 team includes researchers from Kennedy Space Center, Johnson Space Center, Purdue University, and Sierra Space. We are trying to provide the data to support development of plant growth systems for future exploration missions. And the goal of this current study is to assess the impact of two different red to blue light spectral formulations on how well the plants grow and how much fruit they produce, the flavor, the organoleptic acceptability of the fruit, the nutritional composition of those fruits, and the microbial level of the tomato fruit. And we're also going to look at the psychological impact of having plants growing and caring for them, and consumption on the crew members, having the crew members consume the plants throughout the mission. The Veg-05 mission will run approximately 100 days because tomatoes take a while to produce fruit. And this will be the first time that we're going to be, growing these for eating, and we'll have the astronauts pollinating them in space. These plants will be grown also simultaneously on the ground in our ISS environmental simulator chambers at Kennedy Space Center, which mimic the temperature, relative humidity, and carbon dioxide levels of the International Space Station. And we have veggie chambers on the ground here where we will grow them as well. So we will be able to really assess the impacts of growing between flight and ground on these crops. The crew members, after they eat the fruit, will be asked to rate the flavor, the texture, the juiciness of the produce grown under the different light treatments. We expect to have 3 harvests of tomatoes as part of this experiment, and those will be in the spring of next year. And the crew members are also going to be taking surveys to assess their moods and any psychological benefits that growing plants may have when they're living and working in this extreme environment. And this is all very relevant for future long-duration exploration missions. A last kind of fun part of this is we have a mission patch challenge for K through 12 students. This is part of the Fairchild Challenge run by Fairchild Tropical Botanic Garden in Miami. And this is a national challenge where students around the United States are being asked to design the mission patch for Veg-05, and then Veg-05 team members will judge all the submissions, and we will, decide which is going to be the official mission patch based on all of those that students submit. So that's all I have, and I'm glad to take any questions.
Thank you for that. We'll now turn it over to our question and answer portion for Dr. Massa. While we wait to see if anyone has entered the question queue, we'll start with a question. And before I jump into that, I do see we had 2 media entered, so we'll actually turn over to media questions. So first, I'll start with Beth Mund, the Casual Space Podcast.
Hi, Dr. Joy. Thank you so much for this interesting and exciting experiment. For a lot of people who want to follow along from home, can you please expand a little about what kind of tomatoes, again, could we grow from seeds at home, the same kind, and connect by watching them grow on station and growing them in our own kitchens and places? And then please expand a little bit about, are they started as seeds and they contain dirt? Is there little pods where the seeds are encapsulated and they just are placed into a container? Can you describe that a little more for us?
Sure, Beth. Thank you for the question. People can certainly follow along with this at home. So we're growing a commercial variety of tomatoes called Red Robin tomatoes, and you can buy those seeds online. And we're growing these in the veggie plant pillows. So for those who aren't familiar with the plant pillows, these are essentially space grow bags. They're filled with a baked ceramic substrate. It's a calcined clay, and it's porous, so it helps trap water and air into the root zone for the plants, because that's one of the strangest things about growing plants in space is really the behavior of water and trying to water your plants. Water sort of sticks to surfaces and crawls, so it can be really challenging. So this baked ceramic, a porous ceramic product, helps to move and distribute the water to the right places. And we mix it with a controlled release polymer-coated fertilizer, which is a slow-release fertilizer that releases the nutrients that the tomatoes need over time. Just as an aside, that porous ceramic is actually the same material that's on sports fields. It'll be like what you see between the bases on a baseball field, and we're using that in space. And the fertilizer was developed to help prevent runoff here on Earth, so it helps prevent, a lot of nutrients going into rivers and oceans and causing problems. And we've used the same technology in space. So these grow bags are planted with tomato seeds that have been surface sterilized to keep away any bad microbes, and then are glued in place in wicks. They're packaged up, and when they get to the space station, the astronauts will unpackage them, strap them into the base plate of the veggie chamber, and then add water, essentially. And then they keep adding water. Tomatoes are pretty thirsty. They drink a lot of water, so the astronauts will be watering pretty frequently during this mission and hopefully enjoying their experiences with the plant. And then the base plate gets attached to the veggie, which has red, blue, and green LED lights and some fans to move the air. And there's a transparent bellows that allow the astronauts to see the plants growing in veggie as they float by. So we're going to be growing these in 2 different veggie units, and one will be kind of more of a red-rich purple light, and the other will be more of a blue-rich purple light. And we want to see, do the changes in response to those lights, are they the same in space as on the ground? So does that answer your question?
Yes, fantastic. Thank you.
Thank you.
Our next question is from Elizabeth Howell at space.com.
Oh, thanks for taking the time. I was curious about lessons learned that you're applying from past veggies, such as, for example, Veg Pond 2, because there was a lot of discussion back then about creating, future crops with tomatoes and other water-hungry plants. And so what have you been learning from the past veggie experiments that you're using for this one? And what do you hope to get out of this one that you'll be able to do for future veggies? Thanks.
Yeah, that's a great question, Elizabeth. Every veggie experiment that we do, and also our experiments in our advanced plant habitat, teaches us more and more about better ways to grow crops for space. One of the ones that we're actually pulling a lot from for this experiment was our advanced plant habitat chili pepper experiment that ran last fall. So to date, that's the longest plant growth experiment we've had on the space station. It was 135 days, and the astronauts pollinated the fruit in that, we just got a tremendous amount of data. We learned better ways to fertilize as part of that, and we saw the pepper fruit form. So we're hoping that we have as good a production with our tomatoes and veggie. The advanced plant habitat's a much more automated system, whereas veggie is much more manual, and the astronauts have to do a lot more hands-on work with the watering in veggie. The water system in veggie's been a challenge. The ponds experiments are looking at trying to develop less manual watering, and a recent X-Roots experiment by CRS also was looking at different watering approaches for veggie. We are using pillows for this experiment, which we've used in Veg 01 with the lettuce and the zinnias that many people may remember, and also in Veg 03, where we did a lot of different types of leafy greens over several years. And we've refined the watering a bit, the timing of when to water, the use of the reservoir, the root mat that's underneath those pillows. So tomatoes will be a new adventure for us on the veggie team, trying to figure out, how to keep these thirsty plants well watered without overwatering. It's a bit of a tightrope, but we are using all our knowledge from previous veggie experiments, and hopefully we'll be very successful with this one, as we have been with several others in the past. Did that answer your question?
It sure does. Can I also ask how you might be pivoting this for the moon in the future, if you were able to put it out there?
Yeah, that's, it's a bit farther out. We are looking at plant experiments for the moon and at growing, different types of crops for the moon. But at this point, we're not planting the tomatoes yet. I think the development of different crop production hardware, like the Ohalo system, which is currently being developed for the space station, which is more of a larger crop production unit than veggie and the advanced plant habitat, will really help us to move forward for the moon. So that's hardware that's in work. and it will be useful for things like a transit mission to Mars as well as for lunar and Martian surface. When we're on the surface of the moon, we have some gravity. So that makes watering your plants, we think, easier. But obviously, we're going to have to test that out. But we're looking at a lot of different fluid physics approaches to watering plants in these different environments. And then, controlling the atmosphere and the lighting and the temperature, all of those are going to be very equally important. But tomatoes are going to be a great crop for the moon. They're very nutritious. They're very delicious. And we think the astronauts will be really excited to grow them there.
Thank you so much.
Thank you.
And our next question will be from Marvin Marshall at Nighttime News Space Report.
Hi, my name is Marvin Marshall from the Nighttime News Space Report. Thank you very much for taking a question here today. Now, we were wondering if you could talk a little bit more about the LEDs that are going to be used up there. I know you mentioned that a lot of red and a lot of blue spectrum in those lights. Now, why do we see like the blurple lights and not necessarily a full spectrum LED quantum board? So we're just curious if you could talk more about the lights that are being used and everything. Thank you again.
Yeah, sure, Marvin. So Veggie, was launched in 2014 to the International Space Station, and at the time, the LED research and still is very rapidly advancing for horticultural crops. But when Veggie was launched, it has the capability to do red, blue, and then green LEDs. So very, very narrow spectrum. And this, at the time was very common in horticultural lighting. Now, horticultural lighting has advanced, and a lot more lighting now uses white or white with red. Advanced Plant Habitat launched a few years later in 2017, and that hardware actually has red, green, blue, white, and far red LEDs. So it has enhanced spectral capability. And future plant growth hardware for space will have additional spectral capability. But the LEDs are really fascinating because, it gives us the potential to tweak the spectrum to get very specific plant responses. We can grow things under white light, but we can also change the spectrum to try to increase the nutritional content or increase the flavor attributes or the height or decrease the height, and so there's a lot of really amazing plant research out there trying to figure out how you can tune the spectrum to change plant growth. And we're continuing this by using the Veggie as a capability to see how the red-blue ratio impacts the plant growth. In our ground testing, we've seen that plants grown under the red-rich spectrum tend to be taller and fruit a bit earlier than those grown under the blue-rich spectrum. We're interested in seeing if they have different water usage in space and on the ground, and also things like the microbial food safety, because even that can be impacted by light. And while we think that the tomatoes are going to be totally safe, we'll be interested in looking at the microbial communities on the leaves and seeing if those differ from the growth environment. So we're going to do a lot of analysis on the samples that come back and just add to this really great body of knowledge that's forming. Every plant responds a little differently to the light spectrum, and so we want to know more about tomatoes. So thank you for the question.
And we have one question from social media. We are taking Ask NASA questions for this press conference, so we'll turn it over to Allison Tankersley.
The question from social is, when growing Earth plants in zero microgravity, how do they do geotropism? Do you have a fan creating wind so their stems grow strong and not spindly?
Okay, thank you for the question. It's very interesting because there are so many impacts of microgravity on plants at a genetic level, and many of my colleagues who are studying, plant growth look at differentially expressed genes as plants, are grown in microgravity versus Earth gravity. What we tend to see on the crops is if you can get the environment similar between the two locations, plants grow pretty much the same. And so for gravitropism or geotropism, which is, the plant sensing the direction of gravity, that's the number one guiding stimulus on Earth. Roots grow down, shoots grow up. In space, in the absence of gravity, plants key in on secondary stimuli. So for shoots, the big one there is going to be light, and it's really blue light. And so this causes phototropism. So since we have the blue light over the plants, the shoots orient to the blue light, and they grow pretty normally. Roots key in on light as well, and they'll grow, away or towards certain wavelengths, but they also key in on a lot of other things like nutrient composition or water and oxygen levels. And so the root growth may be very different, but we seal it up inside these plant pillows so that they're not too confused, and they can basically find the nutrients and the water levels that they need. So we do have fans. Fans are absolutely required on the space station because in microgravity you have no natural convection, which means the air doesn't, move around naturally. Heat doesn't rise. Gases don't distribute. And since our plants are photosynthesizing and they're taking up carbon dioxide and they're giving off oxygen, we have to have fans to move that airflow around and to bring the fresh atmosphere to the plants and to take the oxygen out to the astronauts. So we do have a fan inside Veggie as well. And so by controlling the environment with the fans and the lights, and by having the astronauts manually control the water, we have the nutrients, we give plants pretty much everything they need to grow in that environment. We have seen with the chili peppers in advanced plant habitat, the orientation of some of the organs was a little different. So the flowers, which are, the reproductive structures, the flowers normally in peppers hang down. But in these, they actually pointed upward. They look like little satellite dishes. And the fruit pointed upward as well. They look like little rockets launching instead of hanging down. So we're really excited to see how the orientation may differ with tomato fruit and flowers. Does that answer that question?
Thank you very much.
Okay.
And our next speaker will be Matt Mickle to talk about iROSA. I'll turn it over to Matt.
Great. Thank you, Josh. So as Dr. Costello mentioned at the beginning of this broadcast, on the next SpaceX flight, NASA is going to be launching two new ISS rollout solar arrays, or what we call iROSAs, to continue the upgrade of the ISS power system. The first two iROSAs were launched and installed last year, and they've been performing extremely well. Each of the iROSAs provide about 10 kilowatts of power generation capability for the station. The new iROSAs will be installed on spacewalks that are currently planned for the 28th of November and the 1st of December. And with a successful installation, that means we'll be about halfway through our upgrade of the legacy solar system that's been on orbit for over 20 years. And with the upgrade, it'll provide additional power for both the scientific payload customers that we're hearing about on this broadcast, as well as commercial elements for the space station. Now, each iROSA utilizes what I think is really an ingenious design. So the backbone of the iROSA structure is a flexible composite carbon fiber boom. And if you can imagine the boom as a soda straw, and you cut it down the middle and then roll up the straw, all the stored energy of that straw, or the boom in the case of the space station, enables deployment without the need for a motor. And why that's important is it increases reliability and also reduces the mass for multiple applications extraterrestrially, such as for satellites and also for future lunar surface infrastructure. The iROSAs use solar cells that are built by Boeing subsidiary Spectralab, and they're assembled and tested on the ground by Redwire, which is a Boeing supplier. Now, incorporating iROSAs into the existing ISS architecture required innovative designs, both for the mechanical attachment to the structures that weren't originally designed with that intent, and also for the electrical integration into the power system. So the designs ended up minimizing the amount of new hardware that was required, as well as maximizing the use of existing hardware and software. These intelligent designs, I think, really showcase the strong engineering capability of Boeing, as well as our adaptability, which is going to be critical as we continue to build out more infrastructure in Earth orbit, as well as beyond to the Moon and Mars. So with that, I'll take any questions.
And as a reminder, if you would like to ask a question, please press *1 on your phone to enter the queue. And while we wait to see if any media have any questions, I'll ask what the performance has been for the first 2 IROSs.
So great question. So far the performance has been slightly better than expected. We did some substantial modeling that takes into account, obviously, the orbits that the space station takes, as well as orientations of the space station on orbit. And the IROSs are performing very well. Again, basically about 10 kilowatts of additional power generation per wing.
And we have had some media enter the queue. And first, we'll go to Elizabeth Howell at space.com.
Hello there. So I realize that probably the spacewalk procedures are still in a kind of an early or moderate stage of planning, but I remember when the last IROSs went up, the astronauts were talking about the challenges of going so far out on the boom, just the time it took, and also, you know, all the other complications that came with it. So do you have any sort of feedback from those spacewalks you're going to be incorporating into the planning process for this time to make it even smoother? Because it did go well, but, you know, other things that you could be improving on?
Sure. Good questions, Elizabeth. So a couple of things that we ran into during the spacewalks, the first 2 for the IROSs, the first 2 IROSs. One was some interference we had while we were unhinging the IROSs to attach to the structure on the station. There was a little bit of interference with the bracket. So we did make a minor change in the design of the slot that bracket interfaces with that we've tested on the ground to ensure that it'll work more smoothly on orbit. And then there was another minor design change that we made for the blanket sunshades, and these are the shading that goes across the longeron, which is the legacy space station solar array structure to prevent thermal loading. So there was a little bit of buckling in some of those sunshades, so we ended up switching to a thinner metallic cable for those sunshades and some additional hinges in the sunshade structure. So again, we've tested those minor modifications on the ground, and both of those we are confident will ensure a better deployment this time. As far as the operations procedures go, though, I think, again, they learned a lot from both the training and the actual on-orbit operations for the EVAs, so we're confident that these will go smoothly as well.
And our next question is from Mark Carreau at Aviation Week and Space Technology.
Yes, thank you very much. Based on the experience you've had so far, and also, of course, in the development planning, what's the sort of estimated operational life of the space station IROSs?
Okay, great question, Art. One thing to point out, I think, about the space station IROSs that's different than the legacy solar arrays is that the capabilities of the Spectralab cells are such that they're more robust with respect to radiation. So while the life of the solar arrays, the legacy solar arrays, we had a fair amount of degradation of power generation capability over the 20 plus years that they have been on orbit. We're going to have a much less steep degradation curve based on the enhanced capability of the Spectralab cells. So as far as lifetime, I mean, we're expecting, capabilities to go well beyond 20 years as far as the capability. Again, there will be some power degradation capability over time, but it'll be much less than the previous generation of arrays.
And our next question is from David Curley at the Discovery Channel.
In kind of vein of question about changes in knowledge, you'll be kind of halfway through. So these arrays, as you just mentioned, can deal with radiation a little better than others. What are you already seeing the next generation? What comes next? What percentage of power increase could you see? What kind of time or lifetime changes? I mean, what has this taught you about the next generation?
A good question, David. So I think, in general, the industry as far as solar cells and solar arrays for spacecraft has been evolving, not just for our experience on the space station, but as well as all the experience within the satellite industry, and frankly also on Earth as far as terrestrial applications. So I know that the teams are always looking at ways of improving the efficiency of the solar cells themselves by minimizing or by maximizing the efficiency. That minimizes obviously the number of cells you would need to have in a solar array to generate the same amount of power capability. And then with respect to the deployment structures, again, I think the fact that we're not just deploying these on the space station, but the rollout solar array concept that Redwire produces is used on multiple vehicles. They most recently, I believe, were part of the DART mission for NASA. And again, looking forward to other potential applications on the moon for infrastructure for that. So it's a continuous process, and we refine both the power generation capabilities as well as the mechanical parts of the deployment mechanisms.
And thank you for that. Next, we'll hear from Myra Nelman-Gonzalez to discuss the Moon Microscope Technology Day.
Thank you, Josh. The Moon Microscope is a portable test kit for in-flight medical diagnosis. It includes a portable handheld microscope that connects wirelessly to any smart device via Wi-Fi, as well as a small contained blood sample staining device. This allows an astronaut to collect a blood sample from a finger stick, place a drop of blood on a slide. You then smear that blood and then stain it and drop the slide into the little microscope, focus it really quickly, and then just take a picture. And so it allows that image then to be collected and transmitted back to the ground, where the flight surgeon can use it to diagnose illness and prescribe treatment. For folks that are not familiar with a blood smear, it's one of the most common universal clinical laboratory hematology tests. So in that blood sample, it'll give information on size, shape, and number of blood cells. And so you have your red blood cells in there. It's what carries hemoglobin, but size and shape gives the clinician a lot of information. You also have your white blood cells, which is what allows you to fight infection. And then you have your platelets, which is what allows you to clot when you have, for example, a cut. So although we don't have a profound clinical problem on the space station, crew members do experience changes in their immune system. So when we go for deep space missions, the stressors increase and our ability to care for the crew is reduced. So that combination really increases certain clinical risks. The project is designed then to create a diagnostic lab capability, a portable lab if you want to think of it that way. It's highly miniaturized and compatible with microgravity and operational constraints. So if you have an ill crew member, they could perform that blood smear and imaging and transmit the images in minutes. So it provides a diagnostic capability for crew members in space or on the surface of Moon or Mars. But it also has other applications with regards to the microscope that you can test the water. Do you have something in there? You can image it off a slide. Also, if you have a surface contamination, or we've even can image regolith. And so overall, the hardware can enable improved medical monitoring, for example, on the upcoming Artemis and Gateway missions. Thank you, and happy to take any questions.
As a reminder for media, press *1 to enter the queue. And while we wait for media to dial into the queue, I wonder if there are any terrestrial spin-offs with this technology.
Yeah, actually, and we've had some feedback. It's the technology can serve as a great tool for the military, for example, as well as medical care in remote areas or in disaster areas, partly with the portability and simplicity of how it's used. So the test kit can also function as a portable lab, if you want to think of it that way, that allow medical staff, and it's simple enough that also a layman can perform that blood smear and stain on a slide, image it, and then actually send those images back to trained people at a hospital, for example, to provide diagnostics as well as any treatments that are needed. Other applications can also be for schools, and we've also had even veterinary medicine as something that has expressed interest.
Thank you. And our next question will be from Marvin Marshall at Nighttime News Space Report.
Hi, thank you for taking another question. Now, my question is about testing regolith in your device here. I heard that you mentioned that is a possibility. Could you expand on that a little more? Thank you.
Yeah, so any sample that can be placed on a slide, we can image on this, and we've done some testing on it, and we were able to successfully image, as long as it's small particles that you can put in there, not necessarily a giant rock. But yeah, the resolution goes down pretty small, so we're able to view that type of sample. Does that
Thank you very much.
Question? Okay, thank you.
Okay. And we'll now hear from Dr. John Hogan on bionutrients too. Dr. Hogan.
Thank you, Josh. It's my pleasure to be able to talk to you today about the bionutrients research we're conducting here at NASA Ames Research Center. Our team is developing microbial biomanufacturing systems to support future space missions. And microbial biomanufacturing is a growing commercial field that is developing the capability to make a wide array of products, such as nutrients, vitamins, pharmaceuticals, plastics, and fuels. Biomanufacturing may end up being an important component of future long-duration missions that involve human habitats on the Moon and especially Mars, because they allow us to make the products there rather than bring them there. For example, one of the major identified challenges of these long future missions is the need for food and medical supplies to have a stable shelf life of up to 5 years. However, studies have shown that the shelf life of certain vitamins, nutrients, and pharmaceuticals is currently too short for these long missions. So we're developing an on-demand microbial biomanufacturing platform technology that can supply these vital health-related products. Our first and ongoing ISS flight experiment, Bionutrients 1, is a 5-year storage and growth microbial biomanufacturing experiment on the ISS. Our team engineered Baker's yeast strains to produce 2 different carotenoids, beta-carotene and zeaxanthin, as demonstration compounds for this test. This approach brings up food-grade media in the dried engineered yeast inoculum within small hard plastic reactors, which are hydrated intermittently by the crew over the 5-year period. The reactors are incubated for 2 days frozen and then returned to us for analysis. And we're well over 3 years into that experiment with positive results so far, and we anticipate that the system should work over the entire 5-year test. We also have what we call stasis packs on ISS, which have a wide variety of different potential host organisms and treatments that are also stored at room temperature for 5 years. They're also intermittently being returned to Earth for analysis and allow us to identify new bioproduction organisms and strains, as well as understand the genetics of these strains and why certain organisms survive longer in space conditions. Building upon what we're learning from Bionutrients 1 experiment, we're now launching the Bionutrients 2 flight experiment. This is improving upon our original system by replacing the rigid plastic growth pack with a very small, lightweight bag. And in addition to our current products, beta-carotene and zeaxanthin, we are also adding the pharmaceutical product, follistatin. That's a protein therapeutic used to maintain muscle mass, which is a serious health concern in reduced gravity conditions. We also have tests to manufacture the fermented milk products, yogurt and kefir. One of the yogurt tests is also producing the engineered product, green fluorescent protein, GFP, to demonstrate using yogurt production as a method to make specific nutrients or pharmaceutical products. We also have a visual dye that changes colors as the yogurt is made to allow the crew to determine when the process is complete. The tests are very similar to the Bionutrients 1 experiments, but we only have 2 runs this time. The first soon after arriving on ISS, and then another at 6 months. These tests will show if the new lightweight bag system provides effective microbial storage and growth of microgravity, and it also evaluates the food safety techniques we've been developing. These results will be used to support the development of our Bionutrients 3 experiment, where we are engineering a single yeast strain to make up to 4 nutrient products at once, and refine the production bag and the safety monitoring systems to allow actual implementation in future missions. And finally, our team is also working to find efficient ways to use local resources, like carbon dioxide, to make the media that's needed to enable large-scale biomanufacturing systems to make bulk products such as plastics, construction binders, and feedstock chemicals. With that, I thank you for your attention and happy to address any questions.
And while we wait for media to join the queue, I wonder if it's possible to scale this process to make larger quantities of products.
Yeah, Josh, great question. So that is definitely a strong desire of us to be able to take what we're producing right now, very small amounts of precious compounds, and we'd like to be able to scale this. It makes sense to use the materials that we can find locally, in situ resource utilization processes, in particular carbon dioxide and water. For instance, on Mars, the atmosphere is about 95% carbon dioxide. There's water in the surface, and there's a little bit of nitrogen in the atmosphere as well. So we have the 4 components of our atmospheric commons where we make most of the products we make here today from. And to be able to use those, we are developing non-photosynthetic methods of converting carbon dioxide directly to organics without using biology to produce the needed organic media to scale up these processes, to be able to make things like bioplastics and binders. We actually conducted the NASA CO2 Conversion Centennial Challenge that was a $1 million prize competition to have people contestants make sugars from carbon dioxide and hydrogen without using biology to forward this. So we have a very strong interest in doing that, and we have a project here we call CO2-based manufacturing, which is using what we're learning from Bionutrients work to be able to scale those processes.
Thank you. And our next question is from David Curley at the Discovery Channel.
But I'm going to need a little bit of help here. Can you give us just a layperson's idea of how this conversion works, and if you have to carry the ingredients that you're going to convert into vitamins or whatever other materials you're looking for, can you carry enough? I'm just trying to get a sense of the process and what do you have to take to make this work?
Great question, David. Okay, so what we're trying to do, particularly with the Bionutrients experiment, is to be able to make compounds that don't have the shelf life for long duration missions. For example, vitamin C, the lack of vitamin C in early seafaring missions killed more people than the wars than they were actually fighting. So that nutrient degradation that is in stored foods has been demonstrated in NASA tests to occur. So we are, and that also includes supplements. So it's both the stored food and the supplements. So many vitamins do well and some do not. And so we are trying to identify which ones do not and are posing this as a potential method to be able to make those small amounts of vitamins, nutrients on demand as needed to supplement either the foods, the stored foods, or if there's some plant growth, something that they can't provide. We do that by bringing up food-grade media. In other words, it's normal bacterial media, except that it's made out of human food components. It's sugar, it's yeast extract, and protein powder. And we bring up the dried engineered organisms, and we are testing to make sure that they can last long periods of time, 5 years, at room temperature, not frozen at -80, which is normal. And then you just add water to that system, let it grow. In that particular scenario, it makes something like it's yeast growing in media, and it smells like bread. And it would be at that point probably heat killed just to kill the yeast themselves. And then it could be consumed. We are also making yogurt and kefir to be able to make those similar products. So we would be bringing, in that case, dried milk powder, which has a very long shelf life, and those organisms, and then making fresh food products that could either be probiotic oriented or medicinal or nutrient oriented as well.
So it's not that you're necessarily reducing the amount of material you're carrying, it's that the material you're carrying will allow you to create something, because it has a longer shelf life than the vitamins, what the astronauts would need for 3, 4, 5 years into a mission. So it's basically, I can't take the vitamins because they won't be any good. I'll take this other thing and I can convert it into vitamins when they need them.
That's correct. Yes, and we're trying to minimize that process to take up as little as possible to be able to make those necessary compounds. And we're using biological methods instead of a chemical synthesis method, which people are also working on that approach as well. Currently, it's fairly large and requires a decent amount of maintenance. So we're trying to go with a very simple system here. But like I mentioned, if we are in a place where we can use the carbon dioxide and hydrogen to make organic substrates, we may be able to make a lot of our media from local resources and not bring it up. So we can either make small amounts of these materials or, again, as I mentioned before, try to scale those systems to be able to make large amounts of bulk compounds.
Thank you.
Thank you, David.
And finally, we'll hear from Martin Nisser from the Massachusetts Institute of Technology. Mr. Nisser.
Thank you, Josh. The Fusion project introduces a new way to enable the rapid in-space manufacturing of modular parts in high volumes. And the reason we're interested in this is that the ability to manufacture structures in space is key to the success of sustainable and long-duration future space missions. The structures that require manufacturing can range from objects at the large scale, such as truss structures for structural support of habitats and solar panel arrays, down to objects at the small scale, such as payload fixtures and tools. To explain some of the motivations behind this project, currently, these objects are largely designed, manufactured, and launched from Earth. But rather than designing these strictly for the operational requirements they need to perform in orbit, the designs for these structures can often be driven by the loads they need to survive during launch, as well as the spacecraft volumes available for them to be stowed in. Also, these structures currently need to be scheduled for launch well before they can be used in orbit, requiring us to anticipate the needs on station before they actually arise, limiting our ability to respond quickly to unforeseen events like breakage or loss of parts. At the same time, there has recently been exciting work developed in the area of in situ manufacturing, using processes like 3D printing that enable us to launch bulk materials that can be launched compactly and then be used to fabricate parts on demand as needed. This not only reduces the burden to adapt parts to mechanical loads and volume constraints during launch, but also allows us to fabricate new parts on demand as needs arise. However, while platforms like 3D printers allow us to fabricate complex 3D parts, these parts are typically limited in size by the build volume of the actual fabrication machine and can require significant times to print. And pragmatically, many of the objects and structures used in orbit can and should be made from modular standard parts in order to facilitate repairs, support replacement and interchangeability, and to limit complexity. In this project, here at MIT and in collaboration with our partners from the European Space Agency's Advanced Concepts team, we've developed a rapid on-orbit manufacturing method based on photocurable parts. At a high level, this method is divided into two processes. In the first process, we identify key geometries and prefabricate negative molds of these as elastomeric silicone skins. You can think of these skins as thin-shelled, flexible balloons, which can be folded down to be stowed and launched at very high packing densities. These are launched together with bulk photocurable resins, a type of polymer that is liquid in its uncured state, which solidifies and hardens on exposure to UV light. In the second process, once reaching orbit, we inject the resin into these skins using hydraulic pumps, which then expand to reacquire the geometries that we initially cast them in here on Earth. And in a microgravity environment, these geometries can reacquire their true geometries without the gravity-induced sagging or warping that would arise in a gravity environment. And lastly, we then irradiate these resin-filled skins with UV light to cure and harden the resin, letting us produce large and stiff monolithic parts in a span of a few hours. To demonstrate the utility of this fabrication process, we'll be manufacturing truss structures that are both sparse and exhibit high aspect ratios. These are geometries that would typically require large storage volumes if they had been prefabricated, or that would require significant printing times or exceed the build volumes of typical fabrication machines if they had been manufactured on orbit using 3D printing, for example. We will also manufacture parts known colloquially as dog bones, which have geometries that make them particularly amenable to mechanical testing. And these samples will allow us to characterize our parts in terms of key properties like strength, stiffness, and porosity, and let us compare our space-made parts against control samples, which we fabricate here on Earth. And we hope with this flight opportunity really to demonstrate that we are able to rapidly manufacture an array of key geometries in high volumes that are not only important for space operations, but also limit the need for precious on-orbit fabrication times and astronaut intervention. And finally, to build a foundation for future work in on-orbit manufacturing across the community and our research teams. Yeah, thank you. Back to you, Josh.
Thank you. We'll now take time for meeting questions. As a reminder, if you'd like to ask a question, please press *1 on your phone. And while we wait, I'll ask what led you to develop an interest in free extrusion and microgravity?
Thanks, Josh. We found that many geometries that are important for space applications are these very sparse, high aspect ratio parts that exhibit high stiffness at low masses, and that these are particularly difficult to stow and launch. And at the same time, we saw that many of the fabrication platforms currently in development turn to additive manufacturing approaches that might require significant complexity and time to actually produce these. So we found that an important unaddressed area was the development of a process that could manufacture these complex geometries using both minimal infrastructure on station and also minimal time, which our platform seeks to do.
Thank you very much for that. And we'll now open it up to media again to ask questions of any of our researchers. If you'd like to ask a question, please press *1 on your phone, and I'll give everyone just a moment in case we have any additional questions. Our next question is from Mark Rowe at Aviation Week and Space Technology.
Thanks. I have two quick ones. For the last presentation, would it be possible commercially to make something in space to be used on Earth using these processes you're talking about, sort of a commercial activity? And then to you, Josh, do you know the payload mass overall for the upcoming SpaceX cargo mission?
For the mass question, I can follow up with you with that closer to the mission.
Thank you.
But I can back to that.
Great. Yeah. Thanks for that question. Oh, sorry. Is that question for me?
Yes, it is. Sorry about that. Go ahead.
Yeah. So it's not been the primary focus of this project to develop geometries that would be used on Earth, but we think that so first of all, the microgravity environment does provide a really rich environment to test the sort of geometries that we're trying to fabricate there, simply by the fact that these silicon skins that we're fabricating and launching to orbit, once they are unpacked from stowage, they will reacquire the original shape that they were packaged in, which they wouldn't do here on Earth. So you can, for example, imagine a very high aspect ratio part, like a pen, for example, which if you had the structure cast as a silicone skin and then you try to inject it with a resin, it would simply sag and turn into a kind of, it would require a kind of bent form, which wouldn't happen in orbit because of the presence of microgravity. And so, to answer your question, it certainly is conceivable that you could fill and cure these parts on orbit and then transport them back to Earth for use. Thank you.
And our next question is from David Curley at the Discovery Channel.
Another question about the extrusions. Did the geometry surprise you? I mean, we know what a beam looks like on Earth. What are the shapes? I don't see any graphics with the post that's on nasa.gov.
Yeah, absolutely. So we can certainly provide some of those if we are able to update that folder. But the kinds of structures we're launching and fabricating now are truss structures which you can imagine as if you've ever seen behind the scenes on a movie set, you see these kind of square cross-sectional truss structures that have these interweaving struts to actually support the geometry. And this is exactly the kind of truss structures that we'll be fabricating in this launch opportunity.
Thank you.
Yeah, please let me know if that doesn't answer the question. Thank you.
And that looks like that's our final question for today. So thank you to everyone for your interest in this important science researching technology that will be launching on NASA's SpaceX CRS-26 mission. You can follow all the research being conducted during Expedition 68 at the space station by visiting nasa.gov/station research news or by following at ISS_research on Twitter. And then you can stay tuned to nasa.gov for more updates on the SpaceX CRS-20 mission in the coming days. Thank you, and have a good afternoon.
Thank you for your participation in today's conference. You may disconnect at this time.